Hi everyone. So I'm just going to get us started with the next part of the lecture. So here we go. Hopefully you're seeing my share screen. All right, the sarcoplasmic reticulum and the T-tubules. We did look at this. I'm just reviewing. That is this network surrounding each myofibril, right? Is that blue one? We'll see it in a second. The terminal cisterns, right, that well form perpendicular cross channels at the AI band junction. So that's the exact location of them when we look at the slide. It's the regulation of the intracellular calcium level. So the calcium is going to be the one that's part of the communication for muscle contraction. And here there's going to be storage and release of calcium. The T-tubules, right, form protrusions deep into the cell interior. They have these functions of increasing the surface area, and they also allow electrical nerve transmission to reach deep into the interior of each muscle fiber. The tubules penetrate the cell's interior at the specific location, which is the AI band junction, and this forms part of the triad, which is that the terminal cistern of one sarcomere, a T-tubule, and the terminal cistern of a neighboring sarcomere form that triad. So this is specifically what the relationship is. So the T-tubule has membrane proteins that are going into that space. These are the membrane proteins are what act as the voltage center. So we're going to have an electrical impulse that is going to cause the protein to change shape, and then this is also going to communicate the need for muscle contraction. The sarcoplasmic reticulum cistern, right? The membranes have proteins that protrude into the membrane space as well, and those proteins control the opening of the calcium channels in the cistern. So an electrical impulse comes by, the T proteins change shape, they cause the sarcoplasmic reticulum proteins to change shape, and this causes the release of the calcium. Okay, so we get an electrical impulse, it causes the T-tubules to change shape, this causes the sarcoplasmic reticulum to release the calcium into the muscle for delivering that information. So the sliding filament theory here, we have basically, let's just break it down quickly. Here's this relaxed state. This is what the actin filament looks like. This is what the myosin filament looks like. And when they're relaxed, there's a space in between, right? And there's a little bit of overlap here. During contraction, there starts to be more overlap. So you can see that well, you can't see it yet in this image. So sorry. So the thin filaments are going to slide past the thick filaments, causing a deeper overlap, okay? And nothing changes shape, everything gets closer together. You get a nervous impulse, and the myosin heads, right, are going to form the cross bridging. So that nervous impulse, right, sent all the information with the calcium changes that we're going to do cross bridging formation. So part one is the nervous impulse coming into the sarcoplasmic reticulum and the T-tubules to transmit that information. Then we get to this step, right, which is that the myosin heads are going to bind. The cross-bridging attachments form and break several times, and that's what causes a ratcheting or a contraction effect that makes those filaments slide next to each other, leading to shortening of muscle fiber. So here it is. This is what it looked like before the muscle contraction or the nervous impulse to contract, and then after everything slid together. So that happened by this ratcheting effect of the cross-bridge cycling, which we'll look at soon. Here's the end result. Everything is moving in towards the center line, which is basically what you can take away from that. Okay. So calcium is the signal, right? And when there's low intracellular calcium, The tropomyosin blocks the active sites, the active binding sites of the actin, so the myosin head can't attach. So if what we're trying to do is cross-bridge cycling, right, in this time of low concentration, there's going to be no cross-bridge cycling. So what's going to happen is there's no area of attachment, so it just doesn't happen. I don't know. This is probably in some slideshow mode where it's going to just move forward without me. Okay, muscle fiber remains relaxed. Good. So when we get the T-tubules to change shape, causing the reticulum to release the calcium, right, into the cytosol, which is the center of the cell, the interior of the cell, which our cell is the muscle fiber or filament. When that happens, that's when everything's going to change, and the tropomyosin is going to no longer block the active site. Oh, I didn't want to read through all of this. So, okay, so you read through all of this. Let me show it to you with the images, so it's not just me acting it out. Skeletal muscle is relaxed until stimulated. Okay, so here, this is when the myosin head is activated. So here, the tropomyosin, because of the calcium influx, has moved itself off of the active binding site, and then the myosin head can bind to it. So step 2, so that's step 1. Step 2 is that there's a working stroke, which means it requires energy. The energy comes from adenosine triphosphate breaking a phosphorus bond, adenosine diphosphate. So and then that other phosphate are released, and that's what gives it a bit of energy to pull the actin filament forward. So that's called the working power stroke. Then so that's what we have here is the working power stroke, and then we have the detachment after. So after the ATP attaches to the myosin, the link between the myosin and actin weakens, and the myosin head detaches. So what has to happen for this to detach is the phosphorus head has to rejoin the ADP, right? So they have to rebind together. And in that action, then there's a release from the attachment site. So then we get back to the cocking of the myosin head, which is just it's like the pre-stroke before it's going to attach to the binding site. And so you had your head detach, and now you have a high energy state where it's ready to attach again for the next cross bridge cycle.
See.
Oh, okay, hold on.
So that's what we'll see here. Sorry, I can't talk.
Okay, so where we were, let me get this back again. Okay, so we looked at the whole, let's just look at this whole thing happening all at once. So here's this is the cross bridge formation. There was a change in the amount of calcium, right, from the sarcoplasmic reticulum, releasing it based on a nervous impulse. It causes a myosin head to attach to the active binding site because the tropomyosin has moved out of the way, so we can where that is unblocked, and then we get this power working stroke, right, where the head is moving backwards and is causing a ratcheting effect. So then we move into the phosphate molecule binding on, creating a higher energy state, and allowing that to release and be ready for forming a new cross bridge. First, to form that new cross bridge, it cocks forward again, and it is able to attach to the site. Okay, so what about when we have a calcium influx? So we talked about how calcium comes in, and there's this phenomena of rigor mortis that basically we get stuck in this ratcheted area. So instead of you need the calcium to move in order to recreate to get the myosin head to detach from the binding site. So if that doesn't happen, which isn't the case when someone dies, then it stays bound in that space. So then you get this rigid, you get a rigid contraction of the muscles, and it occurs about 12 hours after the death. Again, it's this intracellular calcium level releases, and ATP energy is no longer synthesized, right? Because the whole body system is down. And so there's no calcium pumped back into the sarcoplasmic reticulum, which means it's just hanging out there. That is what leads to a permanent cross bridge formation. So because you need it for detachment as well, that we don't have the ATP for that, we can't do that. So it stays in the constant state of contraction. And eventually what happens is the proteins break down and then the myosin releases. So it does, eventually there is, you don't have rigor mortis anymore, but there's a period at about 12 hours and then for several hours of rigor mortis, and then the protein breakdown causes that to detach. So what's organophosphate poisoning? This is something found in insecticides. So when we talk about what we're eating, we don't want this, and it's microdosing this into our bodies, but it's going to bind and inhibit acetylcholinesterase, which is an enzyme that's going to break down acetylcholine. And so we'll look at this again when we see the synaptic cleft and nervous impulses, but I'll just introduce it here as a lack of homeostasis. So we're back at the nervous impulse. So there's a point where the nerve is going to stimulate remember, like around the sarcolemma, the sarcoplasmic reticulum, and start the impulse for the muscle contraction. So there's a miscommunication at that area. So we'll look at this again when you see that. But what happens is that you get spastic paralysis. So when a bug comes onto the food to eat it, then it no longer lives. It gets spastic paralysis. Okay, but we can be poisoned by that as well. That's what hypertonia is. That spastic paralysis. That's what that word means. And it's due to this overstimulation. Botulism neurotoxin is the opposite. You have muscle paralysis due to failure of stimulus, so you have a block happening. The toxins prevent the release of the acetylcholine. So we need to look at the neuromuscular junction to talk more about this. So I'll put it in again. What about tetanus? Tetanus is again at the postsynaptic inhibition. So let's pause with this here. And I'll look at that when I have the action potential nervous impulse lecture going.